Thank you so much, Hanya. To everyone at the Nestle Foundation for inviting me here to be back in wonderful Brazil. It's really great to be here. With a view like that, I might forget that there's an audience here altogether and just stare out into the wonderful Brazil. Let's get started, hopefully. Okay. So I'm going to talk about brain development, and I'm a neuroscientist. So hands up, that's my apology from the beginning, is that I'm a neuroscientist. So that's an important part of it. And I like to start with this story, which is the story of this painting called Le Bateau. And it's a painting by Matisse, and it hung like this in a retrospective exhibition in the Museum of Modern Art in New York for many weeks in the 1960s. And many people came, including Matisse's son, and watched it. And it wasn't until a very eagle-eyed stockbroker called Genevieve Habert came by and noticed that something was wrong. And what was wrong was that the actual painting was hanging... this is going to drag me upside down, and people have been looking at it the wrong way around. And so if you take nothing else from my talk today, just remember that sometimes we need to look at things in a very different way and turn them around. And that's really what I'm interested in relation to brain-gut signaling. So I worked, like many of you, in a medical school, and in medicine, everything goes back to Hippocrates, who is thought to have said, now it could be fake news, but he's thought to have said that all diseases begin in the gut. But when I talk to my psychiatry or neurology colleagues, they would wonder about that. And my neuroscience friends, who are by far the most skeptical of all, often challenge me and say, John, why, if you're interested in the neurobiology of emotions, which I am, why are you so fixated on the gut? But all we have to do is look to our language. We use phrases like gut feelings and gut instincts. We're gutted when we're disappointed. We make gutsy moves when we're brave. We have butterflies in our tummy when we are nervous. So perhaps there is an underlying biology to all of this. And as we've heard already yesterday, the wonderful world of the microbiome has really affected all areas of biomedicine. And the one thing that I want to reinforce this morning is that we are living in a microbial world. The microbes were here first. And so that's really important to remember. And we've heard yesterday wonderful talks about the sheer scope of our microbes. But it's worth remembering that in terms of the genes that we have in and on our bodies, we are more than 99% microbial. And if you think of all of the money that has been spent on the Human Genome Project, it's for less than 1% of our genes. And that's why we're all here, to try and understand this more and more. And some years ago, I got to collaborate with some evolutionary microbiologists to really put together this framework about that we are, as humans, the combination of our human genes with our microbial genes in this hologenome, and that we have to navigate this environmental metagenome. And this has been really important, that we frame things within an evolutionary framework. And once you start thinking like this, then it challenges our whole understanding of what it means to be ourselves in health and in disease overall. And so for brain, we know that across our lifespan, our brain changes, but so does our microbiome. And we heard about the microbiome yesterday, about the early microbiome, how important it is. But also it's worth to reinforce what's going on with the brain at these time windows. And we start to see these critical windows. And that's the other thing I want to reinforce today, is about critical windows overall. And especially in this early life period, there are crucial aspects of brain development that are occurring, and they are occurring at the same time. There are crucial aspects of microbial colonization occurring. And we need to be able to juxtapose these together to understand really the relationship, and that you start to remember that if you mess up something with the microbiome, it may affect the signaling to the brain at these key windows. We've heard yesterday a lot about the different factors in early life that can influence the microbiome, everything from mode of nutritional provision to the environment we live in to exposure to antibiotics. We've been interested in mode of delivery at birth. Do you have a clicker that works properly? Or am I doing something wrong? Because it's really irritating. Anyway, I'll keep going. So the mode of delivery at birth is one of the factors that we've been interested in. And this goes back to early studies from Gloria Domingos-Bello's work showing that infants born by C-section have a different microbiome than those born per vaginum. And everyone has shown Chris's wonderful study that made the cover of Nature. I'm showing it again, but I'm reinforcing not the breastfeeding component, but here the C-section component, which is also very important, that mode of delivery comes through in the Teddy study as well. And it's also not just the missing microbes that's really important, and although we're really interested in these missing microbes, but also it's important to realize that there's blooming of other microbes that shouldn't be there as well. And so understanding what the relative contribution this has to health is really important. And we know being born by C-section increases your relative risk of asthma and allergy and type 1 diabetes, but we know very little about it in the context of brain health. And so that's something that we've been interested in. Initially, we started collaborating with our epidemiology colleagues in Cork, working through various registry data, looking at whether there was some associations, and here the word association is really important. We first looked in the context of autism. We found an association, but it disappeared when we started looking at confounding factors and did a sibling design analysis. We also looked at it in the context of ADHD and school performance, and there seemed to be something there. But as Anya said, I'm an experimentalist, I'm a biologist, so I wanted to look at this in our model system, and a fantastic Brazilian PhD student in my lab, Livia Moraes, developed an animal model of C-section in mice, and what we found was as early as postnatal day 9, we could detect deficits in the microbiome, reductions in bifidobacteria in particular, and this led to enduring effects on brain and behavior, in particular social behavior and anxiety behavior. The good news was that when we put microbes back by a variety of means, including prebiotics and probiotics, we were able to rescue the phenotype. Now this is mice. It's interesting, but then we wanted to do a study in humans. And it was a study we didn't really think anything would work, but we took a group of, I was going to say normal, but Irish medical students, and we stratified them by their mode of delivery at birth, and we bring them into the lab and we stress them. We have very good ethical ways of stressing people acutely in the lab. And that means we bring them in front of these grumpy-looking people in white coats, and they have to pitch their dream job in front of them. Then they have to do a hidden mathematical test where they count back from 2012 in 27s or something like that. And they make mistakes, and we measure everything. We measure their biological responses, their psychological responses. And what we found was individuals born by C-section had an exaggerated immune and psychological response to acute stress. Now, acute stress, that's interesting, but I'm interested in stress-related disorders, so I'm more really interested in traumatic stress or chronic stress. So that comes with a problem. How do we study chronic stress in an ethical way? And that's what's really good about being an educator. We do it all the time to our students. We put them through chronic examination stress. And so we can use the chronic exam stress period. And so we did this, and we also found that there was a slight but significant increase in the immune and psychological response to chronic stress in these individuals. Now, that's interesting, and it may be due to the microbiome, and our animal studies would argue very clearly that it is due to the microbiome, but it wouldn't convince my grumpy neuroscience colleagues, in no way. So we have to come up with more evidence. So how do we come up with more evidence that the microbiome is important in stress and brain and behavior? Well, we look to engineering or other areas of biology, and what people do is if they want to find out if something is important, they take it out and see what happens. And that's what we do when we work with germ-free animals. And the concept of growing up without microbes isn't new at all. It goes back to Louis Pasteur. Here you can see a germ-free man from a sci-fi magazine from 100 years ago. He doesn't look that great. Now, our mice, while they have their own problems, they do look a lot better than he does. But they essentially grow up in a bubble. and they're a really important starting point for us to answer the question, is the microbiome involved, yes or no? But there's no real human translation, with the exception being the boy in the bubble that Paul Simon sang about in the 1980s. But these mice in the bubble have allowed us a really amazing insights into how micro-brain signaling is happening, and have taken me and my lab to places that I never thought we would go to. Where have we gone? Well, one of the places is to the world of myelination. Now, myelin is basically the insulation on which nerve cells require for appropriate conductance. Here you can see two neurons. One is myelinated, and you can see the nerve cell is going very fast. And just like the wires in your house, you need proper insulation all the time. And what we found was, in mice that grow up without microbes in their gut, they have a deficit in genes related to myelination. And when we look at them down the microscope, you can see there's a hypermyelination in the prefrontal cortex, a region of the brain involved in higher cognitive function. And this was a really remarkable and surprising finding for us. We've recently replicated this in the zebrafish model, and it really is telling us that there's something here about how microbial signals are mediating a core process in the brain. Now, hypermyelination doesn't happen that often in nature. It happens in some rare autisms. We mainly think of myelination in the context of demyelinating disorders like multiple sclerosis. And then across a variety of other, and the effects here were much more pronounced in males than females, and that's a common theme we see time and time again. In addition to myelination, we find also changes in the level of the hippocampus, the brain region involved in learning and memory, in terms of new neurons being formed, in a variety of neurotransmitters and synaptic plasticity-related proteins, changes in arborization of neurons across the brain, changes in the brain's immune system, the microglia, and at the level of the blood-brain barrier. And so this has been really, almost everywhere we look, we find specific different changes overall, and that these changes tend to be sex-dependent. And this is another thing where I really have to say, I raise my hands and I don't understand, but there's something about the male brain in early life that makes it very susceptible to microbial disturbances, much more than female brain. Maybe with relates to metabolic syndromes, it's the opposite way around. So, but understanding sex differences is going to be a really important part of this studies as we move forward overall. And here, you don't need to be a neuroscientist to see, this is a neuron from the basolateral amygdala. The amygdala is the brain area involved in fear, in pain, and in social behavior. And you can see very clearly that in a germ-free animal, this amygdala neuron is already in a chronic stress state. It's arborized already. So this was really intriguing for us, because the next thing we wanted to find out, well, what's the functional consequences? And what we found was that there was an increased, there was deficits in fear learning, in visceral pain, and one of the most surprising findings we have in these germ-free mice is when we looked at social behavior. Because mice, like humans, they're very social. So if you put them in touch with another mouse or an empty chamber, they'll gravitate towards the other mouse, but not if they're germ-free. And mice, like, well, maybe some humans, are a bit fickle. So if you give them the chance to have a new playmate versus their everyday playmate, they'll gravitate towards the new playmate. But again, not if they're germ-free. So this tells us that there's something about having normal social behavior and having appropriate microbes in your gut, at least if you're a mouse. All that caveats included. And so a few years ago, together with a zoology colleague and a microbiology colleague, we decided to survey the data that was available across the animal kingdom. And one of the things we find when we put everything together was that whether you're a bumblebee in a hive or a baboon in the wild in Africa, if you start to interfere with the microbiome, the one type of behavior that comes through time and time again is social deficits in social behavior. And vice versa, if you start to interfere with social dynamics and social hierarchies, you start to interfere with the microbiome. And so that's really intriguing for us, for the disorders of the social brain in particular, and offers a lot of potential in terms of where we want to go. And so that led us also to start looking at mouse models of autism, in particular where there is social deficits and we find changes in the microbiome. In particular, we found there was a reduction in Blausia species in a mouse model, and then we put Blausia back. And we were able to rescue a lot of the phenotype in mouse models. And what's really nice to see now is that we're slowly moving towards the human side of things. And so the autism microbiome field is very controversial. This is a really interesting paper which showed that most of the changes in autism in this cross-sectional study were confounded by picky eating and diet. And so disentangling the altered diet is really important in this. And so that was quite controversial. And then another paper last year came out which really collates all of the data that we have right now in the context of microbiome and the autism, and there seems to be some signals there. But as Karen Margolis, who just wrote in Nature Reviews a few months ago, pointed out that we have a long way to go to disentangle the relative contribution of the autistic microbiome with the various factors that are leading to social behavior. But things are moving in the right direction, and that's what I would say. We are getting better and better at understanding and moving away from small studies. This is an example of an intervention study with a specific Lactobacillus reuteri strain, which showed some benefits, not on the core symptoms of autism, but on some of the social deficits that occur in autism. And this is also offering hope, and it's using animal models then to validate that as we move forward. And so really at the critical window perspective, we've been trying to look at this as well. This is studies from our own lab, and we really feel it's really important to juxtapose what's going on in the microbiome and what's going on in the brain at that time window and seeing what the long-term consequences of interfering with that is. And when we talk about the developmental origins of health and disease, the Barker hypothesis, a lot of focus has been on the prenatal environment and the postnatal environment, as well as host genetics. But really we have to put the microbiome as a fourth pillar in this to understand what's going on overall. Okay, so this is William Butler Yeats. He is an Irish Nobel Prize winning poet. In Ireland we don't have too many Nobelists for medicine, but we do for lots for literature. And he won the Nobel Prize over 100 years ago. And he was really fascinated by trying to disentangle the complexities of things into individual components. And so he really, whether it was a chestnut tree or distinguishing a dancer from the dance, is something that he was really fascinated about. And I was able to sneak this into a review in Neuron a couple of years ago, really to reinforce that where we are right now, we are trying to disentangle the entities of how the microbes in the gut are signaling to the brain, and we're only scratching the surface. And that's really important for us, but there are many of these pathways that are really exciting and where we're working very closely with our neuroscience colleagues to try and disentangle these circuits underpinning. One of these pathways is the vagus nerve. We showed many years ago that the vagus nerve is really important for sending signals from the gut to the brain. And when we cut the vagus nerve in a mouse, we were able to block all of the effects of a specific lactobacillus. And so this reminds us that what happens in vagus doesn't just stay in vagus, but will actually affect our emotions in ways that we don't really understand. But it's only one pathway and not the only pathway that we have overall. It is worth reminding you all that the microbiome is like a little factory producing all kinds of weird and wonderful chemicals that our bodies can't produce without it. And so I'm a pharmacologist by training, so part of me is like, can we go to the actual endpoints, to the metabolites? And we showed in our mouse model that if we treat mice, stress mice with short-chain fatty acids, we could actually reverse the effects of stress, so that's kind of cool. And then also in relation to barrier function is another area we're really excited about, and Maria Arborto was a postdoc in my lab, now has her own team, is very fascinated about the relationships between the different barriers, the blood-brain barrier, the choroid plexus barrier, and the gut barrier, and the machinery of how the microbiome interacts with all of them. And we're showing more recently that we can implicate the microbiome and barrier function at the level of the brain, and that short-chain fatty acids could rescue some of these deficits in vitro models. And in terms of the gut barrier, one of the things that we're quite interested about is trying to understand the relationship there. And we talk a lot about leaky gut and what's going on, but we feel that we should build a lot more precision into our understanding of what leaky gut really is. And because the gut isn't really leaky, there is barrier integrity dysfunction, and we need to actually understand what's driving that and how the microbes are interacting that and how various interventions can rescue that. And that's a really exciting area as we move forward overall. So the immune system we've heard a lot about. It's really important as another pathway of communication, and it's one that we're really excited about in early life, especially in the interaction with the early life nutrition. And we've been working now in looking at the weaning reaction and how important weaning is to the immune function, both in the periphery and in the brain. And we feel that this is really an important period that has been neglected, especially in neuroscience overall, because of the evolutionary trends in relation to weaning, and it's something that we will come back to. And then as we move forward, we're seeing human studies starting to emerge, looking at the microbiome in relation to cognitive function. The quality of these studies is getting better. This is a study from Antonio Suarez and Cristina Campo in Granada, really showing that there's a relationship between cognitive function in early life and the microbiome, and they went back into mice to validate these effects. And that's where the translational studies are really important, that we go from mice to humans and we can go back to help validate this, and that's really exciting. And then this is a population study that came out of Sweden, published in the last few months. It is quite controversial because it started off with a large cohort, but it went down as years went by. But these are the types of studies we need to see more of, looking at large cohorts, moving forward, pointing towards looking at brain development and brain function, and that's where the things are moving. We can't forget about the other components of the microbiome. We'll hear from Sasha later about the viruses, but we've been also fascinated by viruses. This is Nate Ritz, who was a grad student who's now working with Sid in Seattle, and we're really showing that the gut virome is really important for modulating gut-brain signaling in the context of stress and perhaps also in relation to social behavior. And that's something that we want to do as we move forward. So the brain and the gut, they may be surprising things to put together. And that's my whole career, has been these two things that really people think shouldn't be put together. And then I was thinking about this, and last summer something happened, was 2 movies were released that you would never put together. But we had the Barbenheimer phenomenon, where you have 2 things. And so it got me thinking that maybe the brain is a bit like Barbie. It's pink, it's susceptible to environmental influences, eludes sex differences, whereas the gut is more like Oppenheimer. It's more longer, it's darker, its main character is wondering why it got there when all it really wants to do is make a bomb. And Cillian Murphy is from Cork, so that's why we like to showcase Oppenheimer in what we're doing. But moving forward, we really have to move towards causality. We really need to move from mouse to human and back again, and really need to get away from just correlation. Here's a correlation study that I collaborated with a group in Bonn, correlating the microbiome composition with cognitive function in young adults, but we really need to move away, because you can correlate almost anything. There is a website which shows you can correlate the composition of you can correlate the divorce rate in the state of Maine in the United States with the per capita consumption of margarine. Now, I am Irish, so we eat like butter. So you can imagine if your partner came home with margarine all the time, it would lead to fights, and this could eventually lead to arguments, which eventually could lead to divorce. And these are the stories we're telling ourselves and fooling ourselves. So we really need to move beyond the correlation. And really, in terms of moving this field forward, we need a lot more longitudinal studies. We need more mechanistic studies. We need to be able to understand what a normal microbiome is and really get at cause and effect as we move forward. But the good news is that unlike our genome, where really all we can do is blame our parents and our grandparents, with your microbiome, you have potential to modify. And that gives people and patients really hope and we need to give hope to people in this world today. So thank you very much. I leave you with that world of hope. We have a wonderful team who come from all over the world to have lots of fun in Cork, including many from Brazil and many from South America, from Chile and Argentina as well. And I leave you with the wonderful words of Hippocrates again, who is thought to have said also, let food be thy medicine. We'll change that to let food for your microbes be medicine for your mind. And then remember that every now and then, just every now and then, it's worth turning things upside down and looking at them in a different way. Thank you very much.
John, thank you. That was really excellent. Think differently, I would say. Think differently. What happens in Vegas doesn't stay in Vegas, and many others. Okay, I know already that there are many questions, and Jose is now joining us for the Q&A session. So I just remind everyone that you can ask questions here in the audience, of course, and those who are joining us online, and I hope though as many people as yesterday that you can ask questions also. So Jose, please. We can start with a Q&A session.
What do I do with this? Sorry.
You have some questions.
Okay. And do I just?
And there are already some congratulations for the very nice presentations. We can only join those people online. I already have it. You want to? Excellent.
I'll say some words in Portuguese because
Should you wish to ask any questions in Portuguese or Spanish, please feel free to ask your questions in Portuguese or Spanish because our translators will translate them into English for them. Feel free to do them in your languages.
Thank you so much for your presentation. It's amazing. We have a lot of questions here. COVID lockdown mimicked germ-free environment, and kids born in that time are having increased diagnosis of autism and other disease. Do you think there is a connection?
Between the COVID, it's too early to say, to be honest. We have to remember also that autism is primarily a genetic disorder and that we know there are environmental components to it. So it's about unmasking what the environmental components are. Only time will tell. A lot of services weren't working during COVID, so we get an increase when you come out of COVID because services were back in functioning. So we have to be careful about how we manage that. But in COVID, we had a living experiment. In COVID, we had a living experiment where we changed social behavior. We all socially isolated, and we also changed hygiene practices in our microbiome. So I think there's a living experiment on all aspects of microbiome brain action to be disentangled over time.
Yeah, and COVID must have some genetic predisposition for the disease because some people had inflammation in lungs and some had in the gut. Sure. In that's different. Is there any evidence that you can bring about lasting microbial alterations to reset your gut to improve psychological outcome? We'll see.
It's a great question. We don't have enough data yet, but we're doing work in adults looking at the impact of fiber and fermented foods in particular. I didn't have time to go into it, but we're hoping that would be through diet would be one approach that would be able to reset and maintain a healthy microbiome, but that's something in adults. I'm also trying to come up with a way, if anyone has any ideas about how you would get teenagers to eat more fiber and fermented foods, but that's a whole other story and would be quite difficult.
Which one comes first? The microbiome dictates the physiological state, or the physiological state determines the microbiome?
Well, both are important, but I always remind people the microbes were there first.
Microbes come first.
Evolution, I mean, we know this is what breast milk is the best example of something that's still outsourced to the microbiome to work on. So,
well, they were here first.
Yeah.
Then we just have 70,000 years or more, and microbes are here for a million years.
Yes, exactly.
I think, Professor.
Flavia Indrio from Italy. On always your talk. I was a little bit, how can I say, curious about this story on boy and girl, male and female, about myelinization. At that time, at that period, around 1 year of age, hormones not yet present, not yet differentiate. Testosterone, estrogen are the same, as you know. So maybe some genetic predisposition combined with some peculiar activity of some microbes. I don't know, there should be something, because as you teach us, it's autism disorder, it is a complex disease. In my points, it's just a misconduct about whole brain, new brain. So there is no connection between hippocampus and cortex, maybe due to this myelinization. And so what is behind this?
No, it's a fantastic question, Flavia. We're trying to figure it out in other systems. So we moved to another model. So we're looking at even in the zebrafish brain, which is a more primitive brain, and we find the same effects. So even at 4 days, old fish have this, germ-free fish have this. So there's something there, and it's probably a combination of genetic effects, But the sex differences in early life in pediatrics are always perplexing. When I talk to my neonatal colleagues, they're always more worried about male infants in the NICU ward than female for brain, especially in brain injury is much more prevalent, it seems to be. There's something about the male brain that makes it quite sensitive, or something about the female brain that makes it protective to these signals. We also have it, I didn't show it, but we have electrophysiology data where we're looking at the hippocampus, for example, and we find very, very clear sex differences, huge deficits in males. Females are pretty okay in germ-free. So we have to figure it out.
There's an interesting question here. Is there a reverse effect? I mean, can stress on the brain change the microbiota? I mean, it's just the nerves of the gut, or the microbiota can be changed by the stress?
Yeah, no, it's a great question, and it's something we're studying. Top-down regulation, both ways. So stress will affect the microbiome, but also there's increasing studies on mindfulness and other types of stress-relieving effects on the brain that will also may have effects on the microbiome. So that top-down regulation is really important as well, and the pathways for that are also being worked out overall on that.
In the future, there is a possibility to change the microbiome of mom and baby to be able to prevent mental diseases or neurodevelopment related disease?
I think that's a bit too far-fetched to think that we could. I think we have to be careful about what we promise people. I think many of these neurodevelopmental diseases have a strong genetic, and by genetic I don't mean heritable. I just mean genetic in terms of the genes are involved, and they can be de novo, or they can be inherited. But I think we have to understand that these are complex disorders of brain development that occur. But I think if we look after our microbes and learn to understand what it is, we can actually have beneficial effects on some of the symptoms emerging. That much I would be confident in saying.
Yeah, nobody becomes sick because they want to be sick.
Yeah.
We become sick because we can be sick.
Exactly.
The genetic that's.
Mario.
Mario?
Okay, right. Thank you, John. Wonderful presentation. Congratulations on your work. Mario Vieira from Brazil. I have a question on, you mentioned that the gut bacteria send signals to the brain through the vagus. Can you speculate on what's the real mechanism this bacteria would send these signals?
Yeah. You mean why or how?
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 Yeah.
Okay. How is actually probably easier than why,
So,
but how we think it's probably through the maybe interactions with the enteric nervous system, and maybe metabolites might be affecting the enteric nervous system, which synapses onto the vagus, and then that's able to signal into the brain. The other part of the puzzle is what happens when it gets to the brain, because it's a neuronal signal, so it's not very complex, and so it lands in the brainstem, the nucleus tractus solitarius, and then it has to find its way to the emotional circuits and various cognitive circuits in the brain, and do that, and that's something we're trying to interrogate now. But people are now looking at this black box. There still is a bit of a black box between lumen of the gut and enteric nervous system, and how these signals exactly are being looked at. But it's probably through microbial metabolites that are at play.
But
Right,
does there need to be a certain level of the metabolites, like concentration, in order to have an effect?
Probably. I mean,
Probably, is what I think. Yes, we don't know probably.
It's very hard to measure,
Yeah.
in situ, what's there.
But it must be some critical mass,
I think
then
so.
eventually you have an effect.
But it also depends on what their target is. So if they're working on a G protein coupled receptor, or they're working on an immune
But
cell.
they're working randomly, I would say.
Perhaps.
Not specifically.
But their target, they have specific targets. So if you look at indole, tryptophan metabolites that make indole, they work on the AHR receptor, which is it has to be present for it to have an effect.
Sites, yeah.
For me, there's a difficult question here, but let's go. What strain of probiotic do you suggest to autism with cognitive social improvement? What the strain of probiotic? Which probiotic you use? Is there anyone?
So I just showed two examples. Data in our published study was a Blautia. We just looked at, and I just showed that paper that's published. And then the study that came in the last few months out of Italy was using an L. reuteri strain. But it's going to be understanding why that strain works in that population at that time for that duration, and I think there's a lot more work to understand, and there's lots of redundancy. It's not just the strain, it's what it's doing and how it's interacting, and so these strains are building up.
Can I just comment on the latter study, because there were two strains, and one of them was Lactobacillus reuteri, and the study was not powered for a clinical outcome. So I think it is promising. It's something to be studied, but we are not there as yet.
I totally agree, Hanya. Totally. It just highlights where the field is going.
Yeah.
And we need a lot more studies with a lot more strains, and with proper outcomes.
Even with the same strains, because we really need to confirm
Perfect.
that the effect is there. Yeah.
There's a question here about schizophrenic children. Could you explain more about the pathway of the microbiome from the typical brain and a typical brain from a schizophrenic one?
Okay. There's less known in schizophrenia, but one of the core symptoms of schizophrenia is social deficits. So we're interested in social deficits across a variety of disorders. So I would hypothesize that the same pathways are at play for the social deficits in schizophrenia as we see in the social deficits in autism, as we've published on the social deficits in social anxiety disorder and in other social areas. For the positive symptoms of schizophrenia, for the psychosis-related ones, we just don't know enough yet data. There's lots of cross-sectional data, but we need proper longitudinal studies to look at it as it waxes and wanes overall.
We have some 2 minutes here for. There's just not a question. That's such amazing speaker, such amazing topic. Enjoyed your presentation. Truly, thank you from South Africa. So,
we join it.
Yeah. Have you seen the difference in women that use antibiotics versus the one that didn't use in C-section in the neurological outcomes?
We haven't studied that ourselves, but other people are looking at that in large cohort studies overall. But most C-sections in Europe will have antibiotics on board, even prophylactically. So it's a difficult study to do. In our animal studies, we have looked at it, and antibiotics can exacerbate some of the effects that we've seen. So we have studied that in animals, but in humans it's more complicated. But in cohort studies, people will be disentangling this.
Jose, there is one question which I would like to ask, and he is here also, and I take advantage that we have 1 more minute. John, if you do not like the term leaky gut, what other term you would prefer?
I think it's not a term, but it's more a concept of disrupted barrier integrity. And I think integrity is a better word than leaky. Leaky gives us the concept that there are holes in the gut. And you know, when I talk to my gastroenterology colleagues, they get very annoyed by this. And I think integrity is really what we say overall. And so that would be a preferable term.
Okay. Another discussion about the terminology. We still have time for one more question. Jose,
One more
please.
question. Well, practically, are we at the stage of responsibly translate epidemiological observations in gut-brain axis to probiotic intervention recommendations? I think
that's a great question to end on, because it tells us that we have a lot done, but more to do.
Thank
Thanks a
you
lot.
so much.
Thank you very much. Thank you very much. It was an amazing presentation.
Thank you.